This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 48, recorded on December 27th, 2012. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. This Week in Microbiology is sponsored by ASM, the American Society for Microbiology, the world's largest membership society for microbiologists. Find out how ASM membership can help advance your science, your career, and your network. Go to asm.org/advance. And speaking of ASM, joining us today from Yale University is none other than the president-elect of that society, Joe Handelsman.
Hi, Vincent.
How you doing there?
Good. Happy holidays.
You too.
Thank you.
So when do you assume the official presidential duties?
July of 2013.
Wow.
In a few days, by the time our listeners hear this, it will be 2013. I'll be able to say July of this year.
That's right. Well, I hope you enjoy that. It's not too much work. It's for 1 year, right?
It's for 1 year, yes. And so far, it's been very exciting. Being the president in waiting is kind of fun because you get to watch all the festivities and excitement, and you don't have to really do anything.
That's right. Like a president-elect.
Mm-hmm.
Also joining us today from Medical University of South Carolina, Michael Schmidt.
Hello, Vincent and Joe. Happy holidays.
Same to you. Although today you're not at South Carolina, are you?
No, I'm in snowy Chicago watching the snow come down. It's always nice to see it snow every now and then and get to re-experience my childhood of shoveling.
Do you have a lot of snow, or is it just dusting?
No, it literally just started about a half hour ago.
And are you going to get some significant accumulation there?
I think this is the lake effect. There was a snow squall off in the lake, and I think it's just blown back. It got warmer because the snow we had all melted on the ground this morning, and the wind shifted, and it's blowing in off the lake. So it's one of those things. But Joe's old location, Madison, I heard from my nephew last night, got something like 12 inches last week during that blizzard, the famous Madison blizzards.
It was a doozy, I hear. It was quite something. They canceled the buses, and all the things that are the last to go seemed to go. So it must have been a pretty bad one to paralyze Madison, Wisconsin.
Wow. Well, here in New York City, we have just cold weather. It rained all night.
How boring.
Yeah, quite. Would you have this rainstorm in New Haven, Joe?
We had snow and sleet, and then it's sort of gradually moving toward rain.
Yeah. Oh, well. Elio is not with us. He's out in the California desert, I guess. That's where he goes on holidays, where they don't have internet. Or if they do, he probably doesn't want to deal with it, so he's on holiday. But nevertheless, we have two papers to talk about with you today. And first one is probably something a bit unusual from what we normally talk about. We usually talk about microbiological sciences, but this has to do with the people who do the experiments. And this is a PNAS paper that came out in October, and Joe is one of the authors. And the title of this is Science Faculty's Subtle Gender Biases Favor Male Students. And so it's great that you could join us to talk about this, Joe, because obviously you're quite familiar with it.
Yeah, I think I've read it a couple of times.
So I wanted to ask you, before we start talking about it, you make a statement in the first sentence that President's Council of Advisors on Science and Technology concluded that training scientists and engineers at current rates will result in a deficit of 1 million workers to meet our demands over the next decade. And so that's a pretty substantial number, yet all we hear about in science anyway is grant problems and people dropping out of the field. So that must be making it even worse, right?
Yeah, it's an interesting paradox that I was quite intrigued by when I first started working on the President's Council project, which was a report to the president on the STEM workforce: science, technology, engineering, and math. And the economists on the working group told us that there was going to be this enormous deficit. And we all kind of looked at each other and said, what you said, that's not what we're hearing. And it turns out that there's a vast gap between PhD and bachelor's level needs. And so we hear about, the PhDs who aren't getting jobs, aren't getting grants, all of those concerns. But the manufacturing industry and the technology industry of the U.S. is clamoring for more bachelor's level trained people in technology and related areas. And when there was a discussion in Connecticut Public Radio about the STEM workforce, they went around to a number of companies and said, do you see this? Do you see this shortage coming? And every single one of them confirmed it and said, it's not coming, it's here. That we don't have enough people to hire who have technical and science backgrounds. So the estimate is that by about 2020, we will be 1 million short unless we really step it up.
All right, so this is not at professor level, PhD level science. This is below that, right?
That's right.
Okay. But they still need us to train them, right?
They certainly do. And I think if we follow the recommendations of the report and we increase training by 10 to 20%, we probably will need more of us. Not a lot more, but some more.
Yeah. So I noticed that all the authors are from Yale on this paper. So I guess this was your idea, or you and your colleagues. How did this come about, this study?
Well, I've been interested in the gender bias and racial bias literature for quite a while. And the kind of bias that seems to be probably the hardest to deal with, but also the most prevalent, is the bias that is called implicit or unconscious bias. This is the unintended kind of bias that most of us are not aware of. And it's not where we're actively discriminating and deliberately discriminating against certain groups. And this is a field that I've read quite a lot in my work on women in science and the issues of why women drop out of science, why there aren't enough women in science, and of course how to meet these workforce needs, because white men only constitute about 32% of the undergraduate student body. And if we don't begin to draw more heavily on women and minorities, we're really not going to be able to meet those needs. And even now, we're not drawing on all the best brain power. And so in reading this implicit bias literature, I've talked about it many times. I've given seminars at lots of universities, and invariably, someone will say in the audience, but we're scientists, so we're not subject to that. And I've always thought, well, that kind of misses the point. This is unconscious and implicit. It's not about being a scientist. And they always argue, but we're objective. We're trained to be objective. And of course, again, it's not about conscious objectivity. These are things that go on below the conscious level. But when that was said to me very emphatically, and the entire field of unconscious bias was sort of dismissed summarily, I recently last year when I worked with a group on diversity at Yale, I thought, maybe they're right. Maybe scientists have somehow trained their minds to be so objective that we somehow aren't subject to those biases that everyone else is. And so I have a postdoc in my group who was working on my teaching projects, and I told her about this interaction, and we kind of looked at each other and simultaneously had the same thought. Well, there's only one answer, and that's to do the experiment.
Exactly.
And so we did, and I happened to be very fortunate of having two colleagues at Yale, who Tori Bruskel and Jack DeVideo, who are two of the really world's experts in implicit bias. And so we had the best people around to help design the experiment. But my postdoc, Corinne Moss-Racusin, who's the first author on the paper, was really the driving force. And she came up with the design and wrote the materials that we used in the study.
So tell us about how this was done.
So we wanted this to be the gold standard of science, which in my mind is the randomized controlled trial. And so Corinne and I talked about how to keep it as simple as possible so that we would have only one variable in the experiment, and if there was bias, it could only be ascribed to gender. And so we developed a description of a student. It was a paragraph description, so we weren't going to ask people to read an entire resume or CV. So we summarized an undergraduate's experience, grades and performance, and just gave kind of a profile of the student. And then we sent that profile with either the name Jennifer or the name John on it to hundreds of faculty across 6 universities. And we pulled out all of the biology, chemistry, and physics faculty from each of those 6 research universities. These were 6 top-rated universities, 3 public, 3 private, and they were geographically distributed across the country. And we asked them, would they be willing to give us an assessment of this student, and asked them a series of questions. And the results, when they came back in, showed that the faculty, if they thought that this description was associated with Jennifer and not John, they thought she was less competent than John. They would be less likely to hire her as a lab manager. They would be less likely to mentor her. And they would pay her $26,000 a year if they were to hire her. And they would pay John over $30,000 a year.
So I was wondering how you worded the explanation in a way that would not tip off the faculty that they were trying to do this kind of a study, basically. Did you word it in a way that made them think they were participating in some other sort of study?
Yes, exactly. And that's something I've learned about social science research from planning this study, and it makes me squirm a little bit because we don't have to lie to our bacteria. And I being a little bit slightly dishonest with subjects. And it's only a brief dishonesty, and you'll see what I mean. We told them that we were developing materials to help mentor undergraduates. And so we wanted an assessment of an undergraduate so that we could use that in our mentoring materials. And there's actually some truth to that, but of course it was not the primary purpose here. And in fact, people fell for it so much that we even had one person start to answer the survey questions and then wrote us this blistering email about how we were dishonest and really just reprobates, because he knew that we were trying to get people to, it was a switch and bait. We were asking questions, but then we were just going to suck them into doing more mentoring, and he didn't have time to do any more mentoring. So
wow.
we suggested that he read to the end of the survey, and at the end of the survey, of course, we come clean and we explained exactly what the purpose of the study was. But at that point, the participants can't go back. It's electronic, and so they can't go back and change their answers. So that's why I say it's a very short period of time of dishonesty, but there is a discomfort about that.
Sure. Yeah. I think that's great the way you put it. You don't lie to your bacteria, although you do things to make them behave in certain ways, right?
I do. I didn't say I don't manipulate them. I just don't lie to them.
That's true.
Well, you're trying to design a binary experiment, and there's a large body of literature that actually supports the notion that people can be influenced by even the most subtlest of clues. And I think the premise here is something as simple as an individual's name and a gender designator on the application. So when I was reading your supplementary materials that actually gives the resume of Jennifer in it, I was wondering, did you think of using as an internal control a gender-neutral name?
We didn't, because the names Jennifer and John have been tested in another study pretty extensively. They tested actually a large list of names, and they found that some names have certain associations and some have others. So, if you pick the name Dylan and the name Tiffany, people have much different images than if you pick Jennifer and John. And so in that study, Jennifer and John came out at about the same place on the scale and at a very neutral level, that the names didn't conjure up anything very distinct. And so the names that come in the middle of this scale typically give the same results. So we could change the name to other female names or other male names and probably have gotten the same result. So that's why we didn't include something completely gender neutral. It's really hard to write also. And so it would probably clue people in, at least who were rating that one, that we were getting at something having to do with gender, because you become very stilted when you avoid the pronouns. The other problem is that it's very hard to get large samples for these kinds of studies. Faculty are busy. They don't want to answer surveys. We all get requests for hundreds of surveys a year probably, and nobody can participate in all of them. And so to have the maximal statistical power, we decided that it should just be a binary study, as you call it.
That was the one thing that intrigued me also about your study. Do you think, I'm always intrigued by sample size calculations. Is a sample size how did you arrive at a sample size of 127 would be sufficiently powered to answer your question? What assumption did you make in order to know that was an appropriate power?
Well, we looked at the size of the difference that we wanted to be able to detect. So we didn't decide on the sample initially at 127. That was the number of responses we got. And we looked at the minimum number of responses we would get to see a certain level of difference. And in this case, we wanted to be able to see a half point or a point on the scale difference as being significant. And we were actually well above the number needed. And what was actually quite surprising to me in this study was how quickly the statistical significance showed up. When we had the first about 13 responses, Corinne stopped by my office and was actually quite disturbed, because she's done a lot of studies of this type before, and she had never seen a difference start to emerge with only a few answers. And she said even at 13, she could see the gender breakdown. And then she got to 26, and she could actually see a statistically significant effect. And that effect just became more and more significant up to the final number of responses, which was 127. So it's an extremely robust result. The social scientists in the group, my co-authors, were all quite struck by how strong this effect is.
So when you gave the faculty this profile, what kinds of questions did you ask them? Did you give them several choices for each question? So would you give them a range of salaries, for example, or was it more a binary, would you hire this person or not?
We gave them a scale. We wanted a little bit more discrimination than just would you or wouldn't you. And so we gave them a scale of how likely they would be to hire. And we also tried to ask some of the very important questions a couple of different ways. So, where the positive in one was equivalent to the negative in the other. And we got very similar results with the different questions, worded different ways, but with the same sentiment. We also gave them a test at the end, which measures what's called more overt kinds of sexism. And there was an interesting association between the expression of overt sexism and the likelihood of showing what we call the implicit bias in this case, because we doubt that anybody was saying, oh, I don't think I'll hire Jennifer because she's a girl. Nothing was that blatant. It's possible, but it's very unlikely. So it was kind of interesting that at least with scientists, you can pick up a certain level of explicit sexism that is hard to pick up these days in other populations. It's not as pronounced as it was 20 years ago, and in many populations not significant at all, but with scientists it is.
How did you get at the mentoring issue? What did you ask them for that?
Oh, simply how likely would you be to mentor John or Jennifer? And that was actually the result that surprised me the most, because forgetting for a minute that it was probably due to completely different kinds of cognitive processes, I thought that the faculty would at least take kind of a paternalistic attitude toward Jennifer, and even if they didn't want to hire her or they didn't want to pay her a lot, at least they would want to help her. And I was quite shocked when there was such a dramatic difference in their likelihood of mentoring Jennifer versus John.
You mentioned that most of the faculty members liked the female more than the male student. I guess that's part of the question that you say shows that there was an overt gender bias, right?
Right, and that was an interesting one because with they did like Jennifer more, but they still wouldn't hire her, and that's always an interesting question to me because people develop these very quick responses, and even though all they've done is read a paragraph, they still have a sense of the person, and just knowing that person was female, they instantaneously had a more positive response than to the man.
Can we pick up on anything about how, to give advice to students, how to increase your likelihood? Now that we know that there is this potential bias, can we teach our female future colleagues how to better write their narratives so that you can get over this bias? Or is that a subject for future study?
Well, I think other studies inform us a little bit about that, but the study itself and the many, many studies in this whole field of implicit bias, which goes back 40 years. I mean, there are hundreds of studies that show similar effects in hiring and salary and promotion. Just about any question you can ask, there will be a male advantage in most populations. And so what we showed and what these other studies show, I think suggests that just manipulating how we present ourselves is not going to overcome the implicit bias. Because in this case, John and Jennifer were presented exactly the same. And no matter how they presented themselves, there would still probably be this intrinsic difference in what I call a certain level of credibility. I think women just have a slightly lower credibility in when we're dealing with this unconscious or implicit type of judgment. There are other studies that suggest that the letters we write for women, the letters of recommendation, have some different characteristics on average. There was a great study out of the University of Michigan where they subjected letters from successful candidates for assistant professor jobs at a large medical school to a linguistic analysis. And so these are all successful candidates, but they found that the women's letters were, first of all, much shorter, 25% shorter than the men's. They referred much more to the women as students and teachers, whereas men were referred to as colleagues and researchers more often. And they were 4 times more likely to refer to the women's personal lives than to the men's. And so there's no judgment here. These women did get the job, so it's not saying that form of letter is a negative, but I think we can imagine how those kinds of references, particularly referring to someone as a student and a teacher when they're applying for a faculty position that's predominantly research, could affect their application. So one thing I think women can do is talk with their mentors about how they write letters, and maybe even share this paper, which is by 2 authors named Trix and Senka. Senka spelled Psenka, and share that paper with their advisors, because I think the more people who read it, the more awareness there will be of how we inadvertently disadvantage women.
I think it's important to emphasize that the gender of the faculty had nothing to do with the bias.
Yeah, that's one of my favorite parts of the study, and one of the things that I think diffuses some of what could be a very tense interaction when we talk about it with men. This is not an accusation against men that they're somehow prejudiced against women. This is something we all do. I mean, all of us. And this is one of the most widespread behaviors that I've seen in the psychology literature. And it's a product of just living in the culture that we live in, and probably goes back many centuries. I doubt that it has changed much. We know it hasn't changed much in the last 40 years, so it probably is a pretty deeply embedded cultural bias. We couldn't find any effects of faculty demographics. We looked at which university they were at, whether they were junior or senior, whether they were biologists, chemists, or physicists, and of course whether they're men or women, and none of those had a significant effect on their answers.
That's amazing. I was really amazed to see that. It's just incredible. What do you do about this? What can be done going forward?
Well, that's, of course, the question we've been struggling with. And there are a very small number of papers reporting interventions, if you forgive the word, that affect implicit bias and reduce it. And so we are actually hopeful that there will be ways of reducing it, either through intellectual processes, like just teaching people about the bias literature, through this paper, for example, our paper. But we're also interested in using artistic approaches. And so we're actually in the process of writing a proposal right now that we're working with the Sloan Foundation on, and that's to look at how film of fictional people in situations that illustrate some of the key findings in bias literature, of how seeing those vignettes on film affects people, as in comparison to people reading the papers or hearing about the results of the papers that those vignettes are based on. And then in a third treatment, we'll actually do a hybrid where we show them the vignettes, and then we have a more intellectual approach where we talk about the data that show that what was in the vignette is a significant problem. And so we're predicting that reaching the unconscious like this, the implicit responses that we have, those sort of automatic responses, may be done more effectively through a more emotional medium, which I think art gives us access to. So that's the approach that we're taking, trying an intellectual and an artistic or emotional approach, and then a hybrid approach, and seeing if we can alter biases.
Do you think that if, I would assume that most of our colleagues, at least in the research field, would be stunned to learn about this and aren't aware of this subtle bias. Do you think just making everyone aware is a good first step? Would that help at all?
I do, and that's why we published the paper and why we wanted it to be in a fairly broad journal and not, in a narrow biology or chemistry or physics journal, because we think it should be of interest to the entire science community, and at least should start some discussion within the science community about whether we apply these biases and how we can avoid them. And there is a little bit of data that simply being aware of biases can actually reduce their impact on decisions.
One of the things that interests me with I know you have to draw a line, but as part of our deficit of 1 million workers, clearly there's a large number of engineers that we're going to need. And I was curious as to why you excluded engineering departments in your sample. Was it just simply the economy of getting the study done?
Well, what we wanted were very equivalent faculty. And it seemed that the biology, chemistry, and physics faculty were present at every university we looked at, and they were similar roughly in terms of the size of the faculty and the type of people that went into those fields. It just in terms of what they do. Most of them were either bench work or theoretical, but they were scientists. Engineers are very careful to distinguish themselves from scientists. And so I agree with you, the engineers are a really important group. And of course, it's an area where women are even less well represented than in the sciences. But there seemed to be more variability in what was called an engineer and where engineering schools fell within these universities. And so just for uniformity, we decided to narrow it to those three fields. And then, frankly, probably for completely selfish reasons, I wanted to be able to talk to my closest colleagues about the research and make it relevant to them. And so since we couldn't diversify to all fields, I wanted to make sure that my field of biology was somewhere in there.
Yeah, it's really interesting. I manage for the Department of Energy and the Department of Homeland Security scholarship and fellowship programs that obviously from the federal government, they are very much concerned about recruiting women as well as underrepresented groups. And so my target audience is often engineering programs, and we have a large number of women in our programs. And I'm always amazed at, I would have thought it would be harder to get, the equivalency to get women and men to apply, but we do indeed get equivalency. So I was curious as to maybe the engineering programs will probably have similar results to what you saw, and they may not be as gender biased as their stereotype would imply from a historic perspective.
Well, I'm a little bit cautious with data about outcomes because we often don't know the quality going in. So I wouldn't make judgments about the implicit bias of the people making the judgments based on apparent parity, similar numbers of men and women, or disparity. I mean, just because there are fewer women doesn't mean there's necessarily discrimination. And if there's parity, it may be that women did as well as men, but they're far more qualified on average. And there's actually reason to believe that that's true, that the women that actually make it in science or engineering have been so highly selected that they are on average better than men, because the people at the lower end tend to be the men because they're selected out of the pool less stringently than the women. So I think it's dangerous to look at the outcome statistics and assume that there's prejudice or discrimination or not.
I think one of the more disturbing aspects of this is the mentoring decision, where most of the faculty are less inclined to mentor women than men, and you mentioned that this can have a dramatic effect on women who decide not to go into a career, because we know that mentors are so important for that.
Yeah, I have to say that I found that a somewhat devastating result, because if you think of all the little interactions, some of which would be called formal mentoring, but others would be similar, like students staying after class and asking questions, or coming to office hours and spending time with faculty, or getting advice on what courses to take or whether to do a research project. And if you think of every little interaction that may be a few minutes or hours of advice or encouragement, if every time women are getting just a little bit less than men of exactly the same quality, you can imagine that by the end of a college career, that adds up to a big difference in confidence. And a lot of men say that they're not mentored and that they don't have role models. But of course, they're either getting mentoring that they don't perceive all the time. Sure. And this would argue that they're probably getting more encouragement in that mentoring. And that really concerns me. Maybe that's having an effect way down the line when women tend to drop out, for example, between the PhD and faculty positions. Maybe it's not the family issues that everyone always points to. Maybe that's a cover, either consciously or unconsciously, for the lack of encouragement, lack of confidence that's resulted from lack of encouragement that they've experienced.
Right. I would be highly suspicious, as you say, of individuals who say they didn't receive mentoring, because just doing these podcasts, all we hear are stories of people who had wonderful mentors and chalk up their careers to that person.
Mm-hmm.
And so I can't imagine anyone doing it. As you say, they probably didn't realize that someone was making a difference in their career.
Yeah, I think that's true. They're just not looking around. And also for men to say that they don't have role models or that they didn't need role models is also kind of specious because that's all men have. I mean,
That's right.
Lots of male role models for them to look at.
Yeah. I just spoke with the chairman of biology here at Columbia who used to be in the theater. That was his life. And then he took a course in biology, and he had a great mentor take an interest in him, and that's why he's in science today.
Wow.
And he says, if it weren't for this mentor, I wouldn't be here, and that's the story of everyone else that I hear. So this is really a really important part of it. And I'm really stunned that they wouldn't want that there's a bias at the level of mentoring. It's just incredible to me. I think everybody needs to read this. It's open access. You can get it, and it's really an eye-opener for everyone. And I think it is important for us to do it here on TWiM. So I want to thank you for doing that with us, Joe.
Well, thank you so much for wanting to cover it. I was really pleased that you wanted the community to hear about it.
Absolutely. And I'm also going to blog about it on my virus blog and just try and reach as many people as possible. Do you give talks about this?
I do, yes. I've given quite a few.
I think that's important that you go out, and I know it's a lot of time for you, but and your postdoc can do as well. It's important because when people hear it firsthand, I think it makes a big difference.
And I think it's partly the discussion that results after reading the paper or hearing about it that is the most important, because that's when people get ideas about how they might be applying bias in their own lives or how they could avoid it.
Right.
I should say, and no discussion about this would be complete without my pointing out that the field of microbiology is one of the best fields in all of science for successful women. Mm-hmm. We had our first women president of ASM in 1929, and we've had a long string of presidents who've been women, and there are many, many prominent microbiologists. And if you look at the percentage of almost anything, any parameter of the microbiology community, we look better from gender equity standpoints than just about any other field of science. So that's just to encourage young women to consider microbiology as a terrific place to be a scientist.
But it's not without gender bias, subtle gender bias.
Of course.
Anyway, right. Yeah. Michael, anything else from you?
Nope. I think it's a very important issue, and it's certainly worth thinking about. And, as you think about papers to do in Journal Club, I would actually commend this paper to do in Journal Club.
Absolutely.
To actually take it apart, look at the science, look at the questions, and then have a guided discussion about how you can think about getting around this bias as you begin to hire people or even select graduate students.
Right. And just to point out how important this is, Nature recently published a feature called 366 Days, Nature's 10 people who mattered this year. And among the 10 is you, Joe, and they call you the bias detective. I'm sure you've seen that one, so we'll put a link to that in the show notes.
Yes, and I think some people who know me saw it because I got a T-shirt called with the bias detective written on it for Christmas. So I think a lot of people have seen that phrase. So
nice.
I won't live that one down too soon.
No, that's a very nice honor, so thanks again. Okay, we have one more paper for you, which is a paper in the Journal of Experimental Medicine, which I find one of the most remarkable papers I've read this year. You'll find out why. It's from a group in Lund, Sweden, at Lund University, and it's called Antibody Orientation at Bacterial Surfaces is Related to Invasive Infection. And I just stumbled across this in my, daily looking at papers for TWiM. I sent it to Michael. I said, is this TWiMable? And he said, oh, yeah, I think this is a good one.
TWiMable?
Oh, TWiMable.
TWiMable. We made a verb. And in fact, that should be a guide to our listeners out there writing papers. Make them TWiMable.
Yeah, we'll pick it up. And so this one deals with streptococcal infections. And in particular, in this paper, it's Streptococcus pyogenes. Lots of throat and skin infections. And more rarely, the bacteria gets into your blood, and there you have mortality. I think 0.6 million invasive cases a year with high mortality rates. And this is the question. Why is this bacterium occasionally able to get into your blood and cause problems? And most of the time it's residing in your oral cavities, and it's not a problem there. So the authors deal with two things that we have to talk about. And Michael, interrupt me at any time.
Yes.
With your corrections and embellishments, of course, because we don't have Elio here to do that. So you can serve in his stead.
Well, the first thing to point out is that there's 700 million strep pyogen infections each year that go around the globe. And if you consider that there's only 7 billion people on the planet, you really can see how big this problem is each year. And so the story that's going to emerge from this paper is really quite remarkable.
So there are two things we have to tell you to start out with. I just picked the number 2, but I'm not sure it's 2. But let's start with opsinization, which is something you might have learned a long time ago. And this is when antibodies bind to a target, in this case a microbe, to help its killing. So an antibody will bind to the bacterium, and antibodies are Y-shaped, and the two Y parts are called the FAB portion, and those are the parts that bind to the antigen. And then the bottom of the Y is called the FC. And when these antibodies bind microbes by the FAB portions, the FC is exposed, and then that FC can bind to receptors on phagocytic cells like neutrophils and macrophages and help to pull the bacteria in. So that's called opsinization. And the other thing is complement destruction of bacteria. That was the second thing. And we all have complement proteins in our blood, and one of their functions is to destroy bacteria, and one way they do so is by interacting with this FC portion of, say, an antibody that's bound to a bacterium. Now, this is a powerful mechanism, this FC-driven stuff. So bacteria have evolved ways to get around it, and there are a whole lot of FC binding proteins in various bacteria. Like we all probably know protein A from Staph aureus because we use it in the lab to capture antibodies because it binds specifically to the FC portion of antibodies. The strep pyogenes that we're going to talk about have M and M-like proteins, and there are others as well. And if you imagine that these proteins are on the surface of the bacteria, they then capture the antibodies via the FC portion. And so they would prevent a phagocytic cell from taking up a bacteria that's coated with antibodies in that way. You okay with that, Michael?
Yep, that's good.
So with that as a background, this paper actually involves a case of a woman who developed a necrotizing fasciitis, which is something we talked about some time ago. She was gardening. She felt a pain in her leg. And then within a few days, she had developed a fever and then a necrotizing infection in her thigh and was brought into the hospital. She eventually survived. But they were able to obtain specimens from her. And in particular, they had cultures from her mouth, her blood, and from this necrotic tissue in her thigh. And they use this really remarkable approach. They use electron microscopy to look at the binding of antibodies on the surface of the strep pyogenes from this patient. And I'm amazed that you can actually see in these electron micrographs not only the fact that their antibodies are little Y-shaped molecules, but you can see how they're binding to the surface of the bacteria. So in the throat from this patient, they're all binding via the FC, and they've colorized their electron micrographs so you can see this. And it's just remarkable. They're all sticking. Now, the FC binding proteins in the bacteria, they form these fibers that come off the surface of the bacteria. So you can see in the throat, the antibodies are clearly binding by the FC, whereas in the blood, the antibodies are clearly binding by the FAB portions. And in the necrotic tissue, they hardly have any antibodies at all present. So this gave them the idea, well, maybe the ability to invade the blood has something to do with the way the antibody is oriented on the surface, and that's what the rest of the paper deals with. But these pictures are gorgeous because you can see not only individual antibody molecules, but of course they've colored them red and green, which is perfect for this time of year, right?
This is true microbiology. This is an N of 1 where they had one patient that they acquired the specimens from. And then the workers here were very clever in that they similarly had mutants of strep pyogens or group A strep. And these mutants that they had were in the M protein and the H protein, which are involved in the binding of the antibody portion, the FAB portion, and the FC region of the immunoglobulin. And the way the authors lay out this story is really so elegant in terms of teaching us that direction is so fundamental to controlling whether or not this microbe will be a commensal, or whether or not this microbe will be cleared, or whether or not this microbe is able to cause necrotizing fasciitis. And how they go about teaching us this story, I think Vincent's going to take us through next.
So the idea is that in the throat, the antibodies are bound to the bacteria via the FC portion. All right. And so that prevents the bacteria from being phagocytosed. So the bacteria can live in the throat and not cause problems. In the blood, the antibodies are binding via the FAB portion, and that exposes the FC and allows the bacteria to be taken up. And then finally, in the tissue in the leg where she developed necrotizing fasciitis, what they found was that most of these proteins, most of these M proteins on the bacterial surface had been cleaved off by a protease, by fibrinogen, basically. And this complex of the M protein with antibodies is known to be inflammatory. So in other words, it induces a influx of cytokines and vascular leakage, and that gives the whole toxic shock syndrome that results from this necrotizing fasciitis. So this was their idea of the sequence of events. And so they then did a series of experiments to test this, which are incredibly elegant. They involve, as Michael said, using strains that lack either one or both of these surface proteins. And they have pooled antibodies from something like 3,000 patients, and they know the exact concentration of antibodies in them. And so they use all of these with electron microscopy and flow cytometry to make a very nice story. So the first thing they find is that the binding of antibodies to the bacteria is concentration dependent. If you have low concentrations of antibodies, they mainly bind by the FC portion. If you have more, you add more antibodies, they bind mainly by the FAB portion. And they think, well, in saliva, there's not a lot of IgG. And so they go to saliva and they show that this is what happens under low concentrations of IgG. You mainly have FC orientation, FC binding to the bacteria. And in the blood, where the concentration is higher, the binding is inverted. The FAB portions are binding. And remember, if the FC part is bound to the bacteria are protected from opsinization and complement-mediated lysis. If they're bound by the FAB, the bacteria can be killed.
And the concentration of immunoglobulin, IgG, in saliva is 10,000 times less than it is in plasma. And so you got 4 orders of magnitude difference in concentration, which then enables the FC portion to effectively interact with the H protein.
Right. So the H protein is crucial for that. When they looked at mutants lacking the H, you don't see this preferential binding of the FC. So H is really important for getting FC binding at low concentrations.
What is H known to do?
H and M are effectively near one another. And what they postulate towards the end is that when the FC binds H, it effectively obscures M such that the FAB domain cannot interact until the concentration of the immunoglobulin is in excess in order to displace, because there's a specific K on and K off. And I don't know, Vincent, did you want to get into the binding kinetics of this?
Sure. We can do that, yeah.
Well, the binding kinetics are very similar to immunoglobulin binding kinetics, and I think H and FC were binding at 10 to the minus 9, and typically antibodies bind down in the, if I'm remembering correctly, down in the 10 to the minus 10 molar range. And so as you increase the concentration, there's always a K on and K off associated with this binding. And the FAB domain does not have as high an affinity. If I'm remembering right, it was 10 to the 6. Does that sound right?
That's
Vincent?
right. That sounds right, yeah.
10 to the 6. So you see that you have a 3 order of magnitude difference, and so the FAB doesn't interact with it, while the FAB does indeed interact with the M protein, which is essential for this fibrinogen network of antibody, fibrinogen, and complement factors to come in and facilitate the opsinization and clearance of the microbe once it's in the serum.
I think these proteins exist as antagonists of antibody.
Of one another.
Yeah.
Yeah, because they're close to one another. And they used some really sophisticated mass spectroscopy showing where these things are binding.
There's a really interesting figure where they take either plasma or saliva, again, from this patient, bind it to bacteria, and then do gold localization of the antibodies. And you can see very clearly that in saliva, the antibodies are mainly bound to the middle portion of this. The surface of the bacteria looks like hair, basically, a thick layer of hair. And these are the M and the H proteins. And these in the saliva, the antibodies are binding right in the middle of that hair, and that corresponds to the FC binding portion of the M and the H proteins. Whereas if you use plasma and you do the same experiments, the black dots, which correspond to the gold-labeled antibodies, are all over the surface, because the FAB determinants can be anywhere on the bacterial surface, right? Whereas the FC portions are
and the
localized.
The difference in the polyclonal IgG concentration is 10,000 mics per mil when it's binding all over the cell, which
Right.
is mimicking plasma. And then the polyclonal IgG at 1 mic per mil is the equivalent of saliva. It's almost literally a straight line across the surface of the bacterial cell, where the FC domain is just literally lining up in almost a straight line.
Yeah, this is amazing. Yeah.
You know, like you can use a Star Trek metaphor of it's creating a shield.
Joe, do you have the paper open in front of you?
I do. I'm looking at the figure, and
So
I'm
look at
just amazed.
Figure 4. Look at Figure 4.
Oh, I was on figure 5. All right, let me go.
Figure 5 is also good too.
Yeah.
So we're doing what Elio hates us to do, but since he's not here, we can do it. He tells us don't refer to figures because the listeners don't have them. But Joe has to see this because, first of all, the lineup of the dots is so striking, saliva versus plasma. Mm-hmm. And if they add more antibodies to the saliva artificially, if they increase the concentration, then the antibodies flip around and bind via their FABs.
Mm-hmm.
And I think that's what we got to remember is antibody-antigen interactions are dynamic. They're not, they just don't clamp on. There's always a K on and a K off.
On and off. That's right. That's really important. Because I think a lot of people think that once an antibody is bound, it's there, but it's on and off. And I love Figure 4D. Look at those. I mean, it looks like someone drew that by hand. It's so clear. The FAB and the FC binding to the green M and H proteins.
It looks like a really badly drawn cartoon, but this is an EM of what's actually going on. And if you think about molecular pathogenesis, of trying to explain molecular pathogenesis, the protein A was discovered in 1960 because it bound antibodies, and it was exploited for the next 50 plus years as simply a way to do co-immunoprecipitations. And the fact that you have this orientation being so essential for clearing bacteria from blood is really pretty remarkable when you begin to think about where it is. And then what happened to this poor woman who was out gardening, she ended up with necrotizing fasciitis, because now, rather than being in plasma where the IgG concentration is sufficiently high, in the tissue we know that IgG doesn't penetrate into tissue as well as it does in plasma. And so consequently, you have a very different effect going on. And they were never able to visualize in the EM the interaction the way they were able to visualize them from the serum and the saliva.
Yeah, that's because in her thigh, all of the M proteins have been cleaved off from the bacterial surface. But what they speculate is that the pain she felt was probably some bleeding in her muscle, and that allowed the bacteria to get in there in a, as you say, it's kind of a protective environment. They don't have the IgG concentration that the blood does, and they took off, basically. They weren't destroyed.
Yeah.
I think this is very interesting because you can see the whole spectrum of her infection in a way, from the saliva to the blood to the thigh, and it all makes sense in terms of this model. Okay, so the idea here is that opsinization and complement-mediated lysis are important. So what they do is they take saliva or plasma and add it to bacteria and then ask what complement proteins are binding. They do mass spec to figure out what complement proteins are binding. And basically they found in saliva there's far less complement proteins binding to the bacteria, as you would expect. Remember, the FC portion is bound to the bacteria. But in plasma, the binding of FAB to the bacteria leaves the FC exposed, so lots of complement proteins can bind to it, including those that are very good at lysing bacteria and opsonizing them as well. Some of the complement proteins are involved with that as well. So again, another piece of the whole story. Not only is the antibody orientation flipped in saliva versus blood, but as you would expect, you have many more complement proteins bound in the bacteria in the blood with the antibodies bound to them.
And so orientation is both important for the innate immune system, the complement, as well as the adaptive immune system. Both arms of our immune system are taking advantage of the orientation in which antibodies interact with a bacterial surface. This is actually going on not only in the saliva, but they speculate that this is actually going on in our guts as well, because in our gut, which is really an extension of our mouths, it's IgA is again the principal antibody, and IgG levels are very low in our gut.
Right.
And so E. coli, of course, one of our major gut flora microbes, has a similar protein to this M and H interaction that we see going on here in strep pyogens.
Right. Now, the last thing they did is they asked, is there a difference in phagocytosis of bacteria if you incubate them with plasma or saliva? All right. So far, they've built this case that the antibody orientation is flipped between the two sites, and that has consequences for complement binding. Now, let's see if it really makes a difference. So they set up a phagocytosis assay using neutrophils, and they can visualize the bacteria being taken up into the neutrophils. when they coat the bacteria with saliva, they barely get taken up into the neutrophils. But if they coat them with plasma, they get gobbled up and destroyed.
And this was in a concentration-dependent manner.
Exactly.
They did a classic multiplicity of infection where they had one bacterium to one neutrophil, and they scaled it up to show you that this was concentration dependent, which was really pretty neat.
Now, the cool thing is they did see some uptake of bacteria when they were coated with saliva. It wasn't zero, all right? There was some uptake, even though there's very little complement in there and there's very little IgG. They were still taken up into bacteria. But what they found was that the bacteria that were opsonized after saliva coating versus plasma lived longer in the neutrophils. So in other words, when in saliva where there's low IgG and they're bound to the bacteria via the FC, they can still be taken up, but they can live inside of the neutrophil longer than they can if they coat them with plasma. So the the bacteria have evolved to be able to survive in the environment of the mouth, where there is low IgG and low complement proteins. They've evolved to make these surface proteins that bind the antibodies in a certain way to promote their survival. And there they live happily, but if they should get into the blood, like you brush your teeth and some of them get in your blood, they're immediately coated by the high concentrations of IgG, and they're taken up into neutrophils or they're lysed by complement. And that's why most of the time these leakages of our normal flora, as they say, Michael, don't pose any problem.
Yeah. Well, you do know that you're bacteremic every time you brush your teeth.
That's right. And just remember that it's your IgG probably that's protecting you, apparently, according to this paper.
And the high concentrations. I was struck by the specificity difference between FAB and FC, that one would bind to that very particular site and they would all line up the way you described. And then the other kind of binds all over. And so does that mean that there's less specificity with the FAB?
Well, it's a polyclonal serum, so it's, the antibodies are...
It's binding to a different spot because it only is recognizing 12 mers.
So the FC binds a very specific sequence, right?
Mm-hmm.
Right.
Whereas the FABs are to different epitopes on the bacterial surface. In fact, they have their double mutant, which doesn't make any M or H protein, and the FAB will still bind because there are some antibodies that bind to different epitopes on the surface of the bacteria. I think that's the interpretation, right, Michael?
Yeah.
So they seem to think this could be a general mechanism of protection against commensals that normally are there and they don't hurt you, but they leak into the wrong place now and then. They have evolved to survive there and not be affected by our immune responses. But if they get into the wrong compartment, that's it for them.
Yep. And complement plays a major role in all this.
Yeah.
Yet again.
Complement and FC. So I thought this was a cool story, and I have to encourage everyone to look at these beautiful pictures, which made me look at antibodies as I had never done before.
Well, this is, again, another good paper for Journal Club because you can assign, one person a figure, and there are 8+ figures if you look at the supplements as well. And it really will hardwire your understanding of how antibodies interact. You can go through the famous scattered plots of how antibodies interact, and you can, play with those equations to understand affinity constants. So this is, again, another really terrific Journal Club paper. Just to review, antibody-antigen interactions. And again, this orientation now is really beginning to help us understand the pathology that we see when bacteria go into the wrong spot, especially here in the case of this woman with the necrotizing fasciitis.
They make the point, which is really worth emphasizing, that S. pyogenes is exclusively human. And so they had to do this study in a person, in samples from a person. And they say because the FC binding proteins in S. pyogenes don't bind IgG from mice or rats. So you couldn't even do this in mice or rats. And so there's no animal model. So they had to use these human specimens. So we have to thank this lady for allowing them to do that. I don't know if she had to give permission or not, but
I'm sure they had human subject committees.
yes.
I find it also fortuitous and beautiful that the cells and the molecules work perfectly when you take them away from the body. You can approximate everything that's going on in the saliva and the plasma just by removing the saliva and the plasma from the body. It's not every bodily fluid that will behave exactly the same in culture.
That's right. All right. Before we wrap it up, let's do a couple of emails here. The first one is from Sierra, who writes, "Hello, Racaniello et al. I am a plant pathologist for a vegetable seed company in Washington. I listen to TWiM, TWIP, and TWiV while I read extensive disease resistance screens. I started out studying microbiology as an undergrad and discovered plant pathology that allows me to study microbes and plants together. Listening to these podcasts keeps my mind engaged while looking at thousands of potentially diseased baby plants. I really appreciate what you all do to put together such intellectually stimulating topics. If I may suggest a topic as well, iron sequestration and uptake in bacteria is fascinating and affects many aspects of bacterial life. I may be biased as I worked on iron uptake during my PhD. I've listened to many of the TWiM podcasts, and iron pops up in discussion now and then. It would be nice to listen to a discussion of this topic from your perspective." Sierra.
Siderophores. What's that famous quote from Kipling about cold iron rules them all?
Is that right?
Yeah, there's the Kipling quote. I'll have to dig it out for the next TWiM.
Okay. What do you think, Michael? I think you'd like to do a TWiM on iron sequestration and uptake, wouldn't you?
Yeah, Gene Weinberg, one of my other mentors from Indiana, would be most pleased if I did a story on iron. It was his passion, and he's written many reviews on the subject over the years of how bacteria steal iron.
All right.
and how important it is. I'll go look and see what I can hunt up.
Yeah.
It's limiting for so many organisms, and it's one electron that changes its availability, the +2 versus +3 form. So it's really a fascinating atom. Definitely worthy of at least one TWiM.
It's TWiMable.
Yes.
It's TWiMable.
It's TWiMable, yep.
The next one is from Eric, who writes, Dear TWiM, episode 2 had a career inquiry from a listener. The listener was considering additional graduate work, but was not sure which way to go or whether to invest the required time and energy in an MBA or PhD. May I suggest intellectual property as a career path? Most patent attorneys are former lab rats. Patent attorneys enjoy a good salary and interaction with some very intelligent and interesting people. Though considerable work is required, intellectual property can be a very rewarding career. And Eric is an attorney in Arizona. I think that's true. I've interacted with quite a few patent attorneys, and that would be a good career for the right person, I think.
Yeah, a number of our graduates have indeed gone into that field. And one of my colleagues who worked in HIV for many years decided to go back to law school and become a patent attorney. And he's now got a very successful practice up in the Washington, D.C. area. And his particular specialty area is in HIV because he worked on it since the beginning. And when he decided to go back to law school, he was already an associate professor, and he decided he'd go and become a patent attorney.
Mm-hmm. Yep.
And so it served him well.
I think it helps to have a good background in science because a lot of the issues do involve pretty hardcore science. So I think it's hard for someone who doesn't have a science background to understand it. All right, our last one is from Ori, who writes, Hello all, quick follow-up from TWiM 47. It seems that many bacteria have means to take up foreign DNA and integrate it into their own chromosomes. This definitely can provide a selective advantage depending on the nucleic acids being taken up. On the other hand, transferring your own DNA to another cell seems to be quite unselfish. For example, if I had some form of an antibiotic resistance gene, why would I give it away to others that would in the end compete with me for nutrients? In essence, what is the selective advantage for giving away nucleic acids? Thanks a lot. Happy holidays.
It's all an issue of timing. Just as today we discussed direction, oftentimes in the first genetic transfer system that was understood, transformation, that's where the bacterium literally dies at the end and gives its DNA to the greater good of the community.
Right.
And then if you're the individual that can take it up, you too may be able to survive. Very altruistic of the transformation class of microbes. Similarly with transduction, the microbe that is being killed by those evil friends of Vincent's, the viruses, slip some genetic information into the specialized transducing phage to give it for the betterment of the population. And conjugation is a peculiar thing because it's all about a selective advantage. And again, microbes are very altruistic, and in fact, I think this was again shown in E. coli, where some E. coli will literally senesce themselves and commit suicide for the greater good of the community.
But the conjugation transfer, I mean, that's basically sex, right?
Correct.
So we have to have sex to have offspring, or that is those organisms that do it that way. And so this is really another version of it in bacteria, right? I mean, you have to give away to produce something. That's the way I would look at it.
But bacteria have an alternative because they can reproduce asexually just by dividing, which is
right.
Typically their most efficient way of propagating themselves. I think conjugation is selected for within a species by its ability to generate genetic diversity and move traits around that can be of an advantage in different environments. But I don't think it necessarily is selected for in the donor organism to be able to donate DNA, for example, to a very distantly related species. So, for example, there's been conjugation demonstrated between E. coli and Bacteroides, very, very different genera. I don't think it's E. coli that was planning to give its DNA to Bacteroides. I think it's important in evolution to think about organisms capitalizing on something that has been selected for other reasons. So Bacteroides is capitalizing, or you might say stealing, the DNA from E. coli, not that there's a selective advantage for E. coli.
So it evolved to help E. coli, but it works with Bacteroides, and so you can't prevent that from happening, basically. Right?
Yeah, I think that's a good way to think about it. But of course, if you think of selection at the gene level, the whole concept of the selfish gene, then I suppose you could imagine spreading the gene around is an advantage for the gene, whether it's in Bacteroides or E. coli.
Yeah. You could look at it that way, right? Genes have their own intent, and but they can't really influence, the conjugation process, I guess. Although maybe they can if they have some positive outcome for the organism.
Right.
It goes back to our discussion about fitness factors from earlier episodes.
Yeah, that there's no intentionality. It's just simply what's selected for, what has an advantage in survival.
Yeah, that's important to remember, that there's no intention. It's always what evolution selects for, right, as being fit for the moment. So I think this idea of giving it away, you have to get away from, because it's what's selected for. And I think what you said makes good sense.
Yeah, I don't think bacteria intend to die and give up their DNA. It's just something that happens, and then other organisms capitalize on it.
Yeah, right. Okay, good question, Ori. I just met Ori, actually, 2 weeks ago. He came to interview for the MD PhD program here.
Oh.
He walked in the door and he said, "I'm a big fan." It's a good way to start an interview, right?
And now we have a whole new world of bias.
Yeah, really. My colleague said, that was a good way of getting in a good word. But, in the old days, there are other ways, too. I didn't let it affect me, of course.
Of course.
I was totally neutral. Anyway, see, he wrote a letter now. That's cool. Because I told him to write a letter to TWiM. And that is TWiM 48, the last TWiM of 2012. And we're moving into not only an odd year, but one with 13 in it. So for those of you who are suspicious, beware. Like every other episode of TWiM, you can find this one at iTunes and at microbeworld.org/TWiM, where we keep show notes. You can look up some of the links that we provide here on the episode. As always, we love to get your questions and comments. Send them to twim@twiv.tv. Joe Handelsman is at Yale University. Thanks for joining us today, Joe.
Well, thanks, Vincent and Michael. It's been a great year for TWiM, and I hope I get to join you again in 2013.
You're always welcome.
Thank you.
Elio was very jealous that he wasn't able to be here with us today.
Oh, yes, absolutely. He had to make a choice between Joe and the desert.
I'll forgive him.
Yeah, don't take it personally.
Nope.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent, and Happy New Year to both you and Joe, and to all our listeners.
Thank you. Thanks for your participation this past year. Look forward to more in 2013. I'm Vincent Racaniello. You can find me at virology.ws. Many thanks to the American Society for Microbiology for supporting TWiM throughout 2012, in particular Chris Condayan and Ray Ortega for their technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.